Hello. This is an educational video that should explain the prokaryotic and eukaryotic cell structure. This is for the IB syllabus for standard level biology only. There are stuff for higher level, however, you might not find all the necessary detail to get full marks on the exam. So for this video, for standard level, it should explain all the things you need to know and do to get full marks on the questions on the cells about cell structure for prokaryotic and eukaryotic cells only. Okay, so the principles, these are things that you should know before watching this video. They are prokaryotics have a simple cell structure without compartmentalization. Eukaryotes have a compartmentalized structure, and electron microscopes have a much higher resolution than light microscopes. These are easy marks in the exams. Okay, so let's get into the basics first. So what is a prokaryotic cell? A prokaryote is a single-celled organism that lacks a membrane-bound nucleus, mitochondria, or any other membrane-bound organelle. So that means everything that they contain will not have a membrane surrounding it. It'll just be floating around in the cytoplasm. An example of a prokaryotic cell would be the archaea. These are found in extreme environments like high temperatures, salt concentrations, or pH. So prokaryotes tend to be bacteria cells because they are single-celled organisms. That is very important, and you should know that. And now eukaryotic cells. So the main feature a eukaryotic cell is any organism whose cell contains a nucleus and other organelles enclosed within a membrane. So that's the main difference. A eukaryote has a nucleus, while a prokaryotic cell does not have a nucleus. That's the main distinguishing feature that you should always mention in an exam when comparing two cells. Also, their organelles, everything within the cell, is enclosed within a membrane, so it's not just floating around the cytoplasm. And eukaryotic cells can be both multi- and unicellular organisms. So an example would be a protista, which is a unicellular organism, or multicellular organism without specialized tissue. Okay, so next you should know what organelles are. These are specialized or organized structures within living cells. These are the structures that carry out all cell activity within a cell. Okay, so now this is a prokaryotic cell. I'm going to be explaining the structure of it and what each structure does, what role it plays within the prokaryotic cell, which are things that you should definitely know, and it might seem like a lot to memorize. That's probably because there is a lot of a lot of structures to memorize and what they do. But once you get it down, you'll be hoping that these questions will come up in the exams. Okay, so first off, the cytoplasm. This is the internal fluid component of the cell. This is where since no cell is compartmentalized, or no organelles are compartmentalized within the prokaryotic cell, this is where all the organelles will be floating around in the cells and all the structures. Okay, so a nucleoid, this is the region of the cytoplasm where the DNA is located. It's not actual, this is what replaces the nucleus in the prokaryotic cell. It's just a strand of DNA, as shown in the picture at the bottom right. This is where all the genetic information is stored. Okay, plasmid. These are circular DNA molecules that may be transferred between bacteria. This is how bacteria reproduce and create an offspring with another bacteria that is genetically different to one another, that is not similar. Okay, so ribosomes. This is a complex of RNA and protein that is responsible for polypeptide synthesis. In prokaryotic cells, ribosomes are 70S. This is the size of ribosomes. And you should remember that, because in eukaryotes there's also ribosomes, but they're different. Okay, so cell membrane. This is semi-permeable and selective barriers surrounding the cell. Semi-permeable means that it only selects a few molecules that allow it to enter and leave. Usually the smaller ones, such as water and other such substances. Okay, now cell wall. So this is a rigid outer layer covering the entire a cell. This will prevent. This is a method of protection, and in the bottom right, this is a diagram of a plant cell. I just want to show you this because the cell wall surrounding the bacteria cell, the prokaryotic cell. So do not confuse both cells together. Okay, a slime capsule. This is a thick polysaccharide layer used for protection against desiccation, drying out, and phagocytosis. Phagocytosis is a white blood cell that is used as part of the immune system for human bodies, and that's how they attack foreign bacteria or cells that could be a threat to the human body. And yeah, so the slime capsule will protect them from that, but not for long. And finally, the flagella. This is a long, slender projection that contains a motor protein that enables movements. Okay, so it's shown in this tail-like figure. That's what the flagella is. Oh yeah, and then there's pili, sorry. This is a hair-like extension that enables adherence to surfaces, attachment to pili, or it also is responsible for bacterial conjugation. It's for so it can reproduce with other bacteria and create a fertile offspring. So these are these small hair-like structures on the slime capsule on the outside of the bacteria. Okay, so now this is this is the diagram of prokaryotic cell. They'll be asking you to draw and label the prokaryotic cell. So this is how it should sort of look like. It could also be an oval shape instead of this rectangular shape. That doesn't really matter. All you need to do is make sure that you label correctly each of the organelles of the cell, and draw them in a way that you can distinguish them. So you must remember the pila or pilas for plural, the cytoplasm, the cell wall. So there's multiple layers. There's cell membrane, that's the gray area. Then there's cell wall. Then there's the slime capsule. You must remember the plasmid, which is just a circle. It's like glycocalyx. That's just a fancy word for slime capsule. Flagellum is just a simple tail. Ribosome, you must mention 7es when it's a prokaryotic cell. It's just a simple dot. And then there's the nucleoid, which is just a simple loop. And yeah, that's how to draw a prokaryotic cell. I suggest you practice it. This will really help to memorize how to draw it. So next, we're going to do eukaryotic cell. This is a much harder cell to draw and to memorize all the organelles because there's just so much more to remember from it. Okay, so let's get into it. First off, the nucleus. This contains the majority of cell genetics, and it is membrane bound. It's not just floating in the cytoplasm, because all organelles now in the eukaryotic cell are embedded with a certain membrane. And this is where chromosomes are kept, and this is where the nucleolus is kept also. So ribosomes. So the structure is two subunits made of RNA and protein, larger in eukaryotes, 80S, than prokaryotes, which are 70S. So remember, eukaryotes are 80S and prokaryotes are 70S. They're just different sizes. Okay, so the function is the site of polypeptide synthesis, this process called translation. You will learn about this further on. All you have to remember is just the site of polypeptide synthesis. So next, plasma membrane. And you don't really need to know the structure because it will never really ask you for structures in the exam. But this is just for safety because the syllabus keeps changing every 2 years, I think. So the structure is a phospholipid bilayer embedded with proteins. Okay, and it's a vital structure, but not an organelle per se. You'll go in much more detail about the structure of the plasma membrane later on. So the function is a semi-permeable and selective barrier surrounding the cell. This vital structure is also in the prokaryotic cell. So next, nucleus. Oh, I already mentioned that. So yeah, it stores DNA, genetic material, DNA such as chromatin, nucleolus, which is the site of ribosome assembly. So endoplasmic reticulum. So the structure is a membrane network that may be bare, that means they refer to bare. They refer to the bare one as smooth ER, or studded with ribosomes, rough ER. So there's two endoplasmic reticulum that you must know. Smooth ER that does not contain ribosomes, and the rough ER contains ribosomes. So the function is transport materials between organelles. The smooth ER transports lipids, while the rough ER transfers proteins. Next is the Golgi apparatus. So the structure consists of an assembly of vesicles and folded membranes located near the cell membrane. So the function of this is it is involved in sorting, storing, modification, and exporting of secretory products, such as proteins or other genetic material. So mitochondrion. So the structure of this, it's a double membrane structure with an inner membrane highly folded into internal cristae. This is the site for aerobic respiration. It's where ATP is produced, which is energy, pretty much. So this is the site of energy for each cell. Peroxisome, sorry. So the structure for this, it's a membranous sac containing a variety of catabolic enzymes. So its function, it catalyzes breakdowns of toxic substances. So this pretty much is, it excretes enzymes, which are catalysts that break down toxic substances or other substances. That's all you need to know. So next will be centrosome. The structure of this is a microtubule organized center, and it contains paired centrioles in animal cells, but not in plant cells. So you need to know. So the function, it's a radiating microtubule from spindle fibers and contributes to cell division, mitosis, and meiosis. These two processes you'll learn in much more detail further on in the syllabus. And then lysosome. The structure consists of a membranous sacs filled with hydrolytic enzymes. The function of this would be the breakdown or hydrolysis of macromolecules. We don't know if it's present in plant cells, but you should just say it's present in animal cells or eukaryotic cells. Yeah, just in animal cells, sorry. And you'll learn about hydrolysis further on in the syllabus. So that's all of the organelles that you need to know for the eukaryotic cells. And it does sound like a lot, but to be honest, all you really need to know is what they do. The structure, you don't really need to know in that much detail as I mentioned it, except for the rough ER and the smooth ER. You must mention that rough ER contains ribosomes, while smooth ER is just smooth, no ribosomes. So, yeah, this is the structure, the diagram that you need to draw. You must be able to draw, and I highly suggest you practice this by drawing it over and over and over again, so you keep it in your memory, because once you do, it'll be an easy 8 or 6 marks. And yeah, so that's the structure for prokaryotic and eukaryotic cells. I implore you to look over this video a bunch of times to get it into your head, all the information that I said, because this is a pretty long video. I'm sorry about that. Or just go to BioNinja. That's where I get all my information. It contains like at least 90% of the syllabus, and you'll find all this information there that I mentioned. So thank you for watching, and yeah.